We are still in chapter 21. This is the very last segment, and it's on immune problems. So we start off by talking about immunodeficiencies. And an immunodeficiency can be either congenital or acquired. And congenital means that you're born with it, acquired means that you get it later in life. And it's going to be a condition that impairs the function or production of immune cells or molecules. One example of an immunodeficiency is severe combined immunodeficiency syndrome. And this is a genetic defect where the person has a deficit in B cells and T cells. It's caused by a defective adenosine deaminase enzyme, which allows the accumulation of some metabolites that are lethal to T cells. It's fatal if untreated. And then treatment is bone marrow transplants. Another example of an immunodeficiency is Hodgkin's disease, and this is an acquired immunodeficiency that causes cancer B cells, and it depresses lymph node cells, and it leads to immunodeficiency. So, severe combined immunodeficiency syndrome would be a congenital defect, and Hodgkin's would be an acquired defect or immunodeficiency. And then we have acquired immunodeficiency syndrome, or AIDS, and this is caused by human immunodeficiency virus, or HIV. And what it does is it cripples the immune system by interfering with the activity of helper T cells. If you remember back to what helper T cells do, they go off and they activate other cells. And this is characterized by severe weight loss, night sweats, and swollen lymph nodes. And then opportunistic infections occur. One example would be Kaposi sarcoma. HIV is transmitted via body fluids such as blood, semen, vaginal secretions. It can enter the body via blood transfusions, blood-contaminated needles, sexual intercourse, and oral sex, as well as mom to fetus. HIV destroys helper T cells, which depresses cellular immunity. Now we're going to talk about autoimmune disease. And autoimmune disease results when our immune system loses the ability to distinguish self cells from foreign substances. And autoimmunity is a production of autoantibodies and sensitized cytotoxic T cells that destroy body tissues. You do not have to memorize this, but I wanted to leave this up here because there are some examples of autoimmune diseases. Rheumatoid arthritis destroys joints. Myasthenia gravis impairs nerve muscle connections. Multiple sclerosis destroys white matter myelin, which means it's going to affect the nervous system. Or myelin covers like neurons. Graves' disease causes hypothyroidism. Type 1 diabetes mellitus destroys pancreatic cells. SLE affects multiple organs, and glomerulonephritis damages kidneys. Treatment of autoimmune disease includes just suppressing the entire immune system. This can be done with anti-inflammatory drugs such as corticosteroids, in addition blocking cytokine action and blocking co-stimulatory molecules. Some of the research into the treatment of autoimmune disease includes activating those regulatory T cells, which, remember, kind of suppress the immune system response, inducing self-tolerance using vaccines, and then directing antibodies against self-reactive immune cells. So autoimmunity can be caused by a couple of different things, but we say that your slide here, your book says that it's caused by weakly reactive lymphocytes that are activated by. So we have foreign antigens that resemble self-antigens, and basically these are going to be the issue is going to arise because antibodies that are produced against a foreign antigen may cross-react with the self-antigen because it closely resembles the self-antigen. In addition, there could be appearance of new self-antigens, and these could be generated by gene mutations, changes in self-antigens by hapten attachment or infectious damage. Remember, haptens are incomplete antigens, and they attach to body cells. And at that point, the immune system goes after not only the hapten, but the body cells as well. And also, there could be a release of novel self-antigens by trauma to a barrier. So, first of all, I'm going to define novel would mean new. And what this means by trauma to a barrier is there's some parts of our body that our immune system doesn't reach. For example, parts of the eye. So let's say you had trauma to your eye and it somehow pierced a barrier. And at that point, it introduced new or novel self antigens into your bloodstream that could trigger autoimmunity. All right, now we talk about hypersensitivities. And after this, we'll talk about development, and that's it. Or I guess, development and aging, and then that's it. So we have hypersensitivities, immune responses to perceived threats that cause tissue damage. And these perceived threats are otherwise harmless. So a perceived threat, an example of a perceived threat could be something as benign as pollen. There are different types that are distinguished by their time course and whether antibodies or T cells are involved. Antibodies cause immediate and subacute hypersensitivities, and T cells cause delayed hypersensitivity. Immediate hypersensitivity is also called acute or type 1 hypersensitivities or allergies. They begin within seconds after contact with an allergen, and the allergen is just the antigen that's causing the allergic reaction. Now, the initial contact with that allergen is asymptomatic, but it sensitizes the person. So let's say, for example, you've decided you want to try new foods with your child, and you give your child something like peanut butter. Well, the first time and let's say the child is going to be allergic to peanut butter. So the first time, all it's going to do is actually sensitize your child to peanut butter, but the child's not going to have an actual reaction. What's happening is activated IgE. Remember, this is the antibody that is involved with allergies, allergy IgE. So activated IgE against the antigen binds to mast cells and basophils. Subsequent encounters with that same allergen cause a flood of histamine, which is going to induce the inflammatory response. Histamines are going to cause vasodilation, leakiness of vessels, which is going to lead to things like a runny nose, itchy hives, watery eyes. If the allergen is inhaled, it can cause asthma. And then antihistamines are needed to control the response. Allergic reactions can be either local or systemic. Local reactions involve mast cells of the skin or respiratory or gastrointestinal mucosa. And a systemic response is known as anaphylactic shock, usually seen with injected allergens such as a bee sting. Bronchioles constrict, making it difficult to breathe. And then vasodilation results in low blood volume, which could cause circulatory collapse, and treatment is epinephrine. Just speaking based on my own experience with allergies, I am very, very allergic to raspberries. Back in high school, which was a long, long time ago. So we're now talking 20 something years ago, almost 30 years ago. I found that I was allergic to raspberries, and the first thing that happened was I got a rash. That was it, a rash. Subsequent exposures to raspberries oftentimes was accidental, but eventually it led to my throat closing up. So your response to allergies can actually get worse over time, or your response to allergens, can get worse over time. So let's look at this. So we have the initial response to the allergen. So we have the allergen here is going to cause these plasma cells to release IgE against that particular allergen. The IgE binds to mast cells and basophils, and these guys are going to be circulating around. Subsequent exposure to the same allergen causes the allergen, well, the allergen will bind to the IgE on the basophils and mast cells, causing a release of histamine. That histamine is going to cause blood vessels to dilate and become leaky. It's also going to cause the secretion of a good bit of mucus and constriction of small respiratory passages. Excuse me. Now we have subacute hypersensitivities caused by two other antibodies, IgM and IgG, transferred via blood plasma or serum. There's a slow onset, long duration. One of the types of subacute hypersensitivities is a cytotoxic reaction or type 2 reaction. Antibodies bind to antigens on specific body cells, which will stimulate phagocytosis, as well as complement-mediated lysis of cellular antigens. An example here would be mismatched blood transfusion reaction. And then we have immune complex or type 3 hypersensitivity. This is where antigens are widely distributed in the body or the blood, and large amounts of insoluble antigen antibody complexes form, and they cannot be cleared from a particular body area. This causes intense inflammation, local cell lysis, and cell killing by neutrophils. An example of this is SLE. And then finally, we have delayed hypersensitivities or type 4, slow onset. The mechanism depends on helper T cells. And then we have cytokine activated macrophages, and cytotoxic T cells cause the damage. And an example is allergic contact dermatitis, like you get whenever you're exposed to poison ivy. So finally, we have developmental aspects of the immune system. Immune system stem cells develop in liver and spleen in weeks 1 to 9. Bone marrow after that becomes a primary source of stem cells, and then it continues throughout adult life. And lymphocyte development continues in bone marrow and thymus. As we age, the immune system begins to wane. We have greater susceptibility to immunodeficiency and autoimmune disease, greater incidence of cancer, and why the immune system fails is unknown, but it could be due to the atrophy of the thymus and decreased production of naive T and B cells. Those are your questions. That's all I have for you.